Today I'm really excited. I'm going to be sharing with you guys a project that I've been working on since the pandemic, basically. So I'm the coordinator for the Biology 101 labs, and then I also teach a section of Biology 101 lecture as coordinator for the Biology 101 lab. And due to the pandemic, we lost a lot of that hands-on learning that you would normally get from a lab course. And so to overcome this obstacle, what I've been working on is making these custom lab kits that students can pick up or we can ship to their house, and they can complete this exercise with their TA virtually. We actually, for this semester, are piloting 2 labs that will be custom lab kits. The first one is the pH and buffers lab, and then in a couple weeks, we're going to try a photosynthesis lab kit. I want to quickly give a little bit of background about pH and buffers just for people that are not in biology or in chemistry, so we can have a little bit of a background. So the pH scale is a really powerful tool for scientists and allows us to measure the concentration of hydrogen ions, or H+, in a solution. And so you can see our pH scale here. We have the pH starting at 0, and it ranges from 0 all the way to 14. And so solutions that are acidic would be ranging from 0 to 6, and these would be having a high hydrogen ion concentration. When solutions are neutral, they're going to have an equal concentration of these hydrogen ions and the hydroxide ions. And then finally, when a solution is basic or is a base, it's going to have a low hydrogen ion concentration. I want to point out here lemon juice. So you can see this red star up towards the top. It has a pH of 2. So this solution is very acidic and it has a high hydrogen ion concentration. So this is going to be the acid that we're using in the lab for today. The second thing I want to go over are buffers. So a buffer is a substance that's going to minimize the change in the concentration of hydrogen ions or hydroxide ions in a solution, so that when you're adding an acidic or basic solution to a buffer, the pH is going to remain more or less the same. And the way that buffers work is that they're able to donate or accept hydrogen ions to maintain this pH, or to help maintain a fairly close pH to the original. So the example that we're using for this class today is bicarbonate. And what you can see is it's able to donate hydrogen here, but it can also accept a hydrogen ion here. Now that we've gone over the acid, which is the lemon juice, and then our bicarbonate buffer, I want to go over our experimental design here. And so what we're going to be looking at is a bicarbonate solution, and we're going to be looking at the change in pH when we add drops of lemon juice to our solution. So just to give a refresher, the lemon juice has this high hydrogen ion concentration, and the bicarbonate buffer is going to accept these hydrogens and prevent the pH from decreasing or becoming more acidic. And so at this point, once we've given the students, a background of this, it's usually a lot more in depth in class. We would then go over our objectives, and the students would create a hypothesis. And so the 2 objectives for this lab are for the students to determine the pH change of water and a bicarbonate solution after the addition of drops of lemon juice. And then we also have the students identify an unknown solution. So we'll give them a vial that says unknown A or B, and one of them will be a water and one will be a bicarbonate solution. So at this time, we usually would stop and have the students create a hypothesis, and we want it to be based around this first objective. And so an example, you can say, upon the addition of drops of lemon juice, the pH of water will drop steeply, whereas the pH of the bicarbonate will remain fairly consistent. So once the students have created their hypothesis, we then would start getting into the lab kits. And so students have already picked these up, and the TA will walk through what's in these lab kits. So they have 3 glass vials, 4 plastic pipette droppers, 4 plastic tubes that contain 4 different solutions. So one being water, another being the bicarbonate buffer solution, lemon juice, and then an unknown solution. And then last, we give them a strip of pH paper so that they're able to measure the pH of their solutions. So the first thing the students are going to do is generate a table that looks very similar to this. So you can see across the top, we have the number of drops of lemon juice, so ranging from 0 all the way to a total of 20 drops. And so at 0, they can measure their pH of their solution before we add any lemon juice. So this is kind of their baseline control. And then in the rows below, we have our water, buffer, and then the unknown solution. And then the students are going to take their long piece of pH paper and break it into 18 smaller strips, and they're going to place these on each of these white boxes. In step 2, they're going to obtain their 3 glass vials and label them as water, bicarbonate, and unknown. And then they're going to add 5 mL of each solution and put it into the appropriately labeled tube. So now what we're going to do is measure our initial pH of these solutions, and what they'll do is pipette and place a few drops of their solution onto the strip of pH paper and put it into this first column here. So this will give them a baseline to see what their pH is at the start of the experiment, and then they can see the changes that are occurring as we add more and more drops of lemon juice. And then in step 4, we are going to start adding drops of lemon juice. So if you recall from the table before, it started off with 2. So they're going to add 2 drops of lemon juice into each of the glass vials. Mix this really well so it's well distributed in their solution. And then they're going to go back to their table and measure the pH again by placing a few drops of their newly mixed solution onto the pH paper. So then they would move back to their table here and then put the drops in the second column here. So this process, steps 4 and 5, would continue over the next 3 or 4 more times. So they would then add 3 additional drops of lemon juice into their solution to have a total of 5 drops of lemon juice, and then they would measure their pH again. So this would repeat until they've reached a total of 20 drops of lemon juice. And so what's nice about this is that these pH strips of paper are going to change color to indicate what the pH of that solution is. So they're able to visualize and see what is happening to their solution as they're doing it. At this point, they're going to want to now compare their colors and match it to the pH in this legend here. So this comes from the company that we purchased the pH paper from. And so what you can see is that each of these colors are indicative of the pH of the solution. So the pH paper is going to change in response to this. So they're going to compare the colors that they observe in their table and write down a value for each of the 18 squares in their table. And then they're going to collect this data and create a table. So you can see now we've shifted this into, this is just a screenshot from Microsoft Excel, and they have their drops of lemon juice, and then they're going to be measuring the pH for all 3 of their solutions. So this will look like a variety of numbers that are put in based on the legend from the previous slide. And then finally, which I think is the fun part, is they get to put this data and create a scatter plot. So now they can actually visualize their data, and then they're able to tell if their data supports their hypothesis or not. So they can now answer that question like, yes, this data supports my hypothesis, or no, the results actually indicate blank. And then they can distinguish what their unknown solution is here. So I just want to go over a couple of the techniques that are used and some things that they can gain from doing this virtual hands-on lab exercise. So it helps them to become more comfortable with performing experiments. So for a lot of students, this might be their first time doing a lab. If they didn't have the chance to do it in high school, or this might be their first lab in college. So it can be a little bit intimidating to do some of these experiments. So this one teaches some really, really basic laboratory skills, such as using pipette droppers, measuring accurate volumes, determining the pH of solutions and comparing that to a legend. And then they also get to collect data and create a table and figure in Microsoft Excel. So we really do get to walk through the scientific method in this lab, which I think is really fun, while also letting them do the exercise on their own, even in a virtual environment. So that's all I have to share today. I'm happy to take any questions or suggestions. I'm always open for anything like that. Thank you.